Hello, ni hao, namaste, bonjour, guten tag, vanakkam, habari. Welcome to all of you. My name is Bali Palendran. I'm calling from Stanford University in California, and it is my distinct honor and pleasure to welcome and introduce our guest speaker this morning, Professor Maria Mota, who is the Executive Director of the Institute for Molecular Medicine in Lisbon, Portugal. Now, many of you know Maria is a pioneer in the field of malaria pathogenesis, understanding the molecular mechanisms by which this malaria parasite interacts with our host defenses. She has a very interesting career path. She grew up in Portugal, obtained a degree in biology and a master's degree in immunology from the University of Porto. She then did a PhD in molecular parasitology from University College London. Then she moved to the United States, where she did her postdoctoral work in the famous lab of Victor and Ruth Nussensweig at New York University. It was there that she began her pioneering work on malaria pathogenesis. I think she's going to tell you a little anecdote of how the first paper she published in Science came about through a perfectly random chance encounter. But she's really done a remarkable series of studies in malaria pathogenesis. For example, in a beautiful paper in Science that I mentioned, she showed that the way that malaria parasites infect a hepatocyte is not directly, but rather like a bullet going through multiple walls or multiple cells before they find their ultimate destination. Remarkable observation. She's also provided many exciting clues about how the immune system interacts with the malaria parasite. So as you know, malaria parasites have many tricks up their sleeves. They can evade the immune system in many different ways, like suppress dendritic cell function and so on and so forth, and suppress, evade autophagy. This has all been a series of seminal observations from Maria's lab. So her pioneering work has been recognized by many awards and honors, including the Young Investigator Award from EMBO, her appointment as the Commander of the Order of Prince Henry, which is a national honor of Portugal, the Premier Pessoa Prize, the Pfizer Prize, and the Sanofi Institute Pasteur Prize for mid-career scientists. So Maria, we're truly honored to have you with us. But before you begin, as is customary, we have to ask you a question. And this question, if I may, I'd like to ask you, could you share with us one of the most impactful decisions in your scientific career?
So what I bring is something that didn't initially has nothing to do with science, but then became science. So in 99, yes, I'm that old. So I was at that time living, the previous century, I was living in New York, and I was working at NIU in Victor Nussensweig lab. And I was living in a building, in an apartment within a building that was from NYU. And one day I entered this elevator to go to my floor, and there was the person inside that I have seen in the department of parasitology before. Was a new person in the department, never spoke to her before. And this was a woman, and she was a little bit, she was not crying, but you could see that she was sad. And I fighted, so the door closed, and I did fight because I'm Portuguese. I was educated in a Portuguese background that, of course, the new generation is becoming different, but in my generation, everyone would ask everyone about everything in their lives. Okay, but at the same time, I just left London, and in Britain, it was very different. People would not ask any personal questions or anything. So I was fighting on that moment, like, should I ask her if everything is okay? Maybe I should not interfere, anything. And so when the door opened for me to go to my apartment, she was staying inside. And at the last minute, I decided to ask her, are you sure everything is okay? I don't want to interfere too much, but, if you have a problem, just knock in my door or something like that. My apartment is such and such. And in fact, the door was closing, but she open again. And she said, oh, in fact, I would love to have someone to talk with. And so we went for a coffee. And within this coffee, I realized that she was very upset because she was not allowed. She was a young group leader starting a lab, but she realized what was promised was not there anymore. And she was upset and she wanted to work on a different parasite that was Trypanosoma cruzi and hood healing. And it happened that the day before I just saw a movie and realized that probably the malaria parasite was making some wounds in cells, was making some holes. I said, you see, are you an expert on healing? We should talk because I really saw this. Next day, we decided to go to see this movie again, the two of us together. And 3 years later, we were publishing this paper in Science showing that the parasite doesn't invade the first hepatocyte that it encounters, but really, traverse several cells by causing, some holes or, disrupting peristaltic vacuum, many aspects here, but causes a disruption of the plasma membrane. And we were the first ones to prove that. And of course, has, different implications. And these days, my lab is still having a project that we are working on the implications of this. So really, after the pandemic, especially, I think this is very important to think about, that sometimes, we just need to go out of our house. We need to talk to people. We need to communicate, because sometimes the best ideas come from, the less expected moments of connection with other human beings. So that would be my message, my main message.
What an inspiring message, Maria. Thank you for sharing that with us. That's truly inspiring. And it's a reminder to all of us that, in fact, one has to keep an open mind, and you never know where your next brilliant idea might come from. It might come from your neighbor who's walking his dog for a walk, right? But anyway, without further ado, before you begin, I should also mention, as is usual, we will not be having a formal Q&A session. But if you do have questions, please do feel free to tweet these to Maria. Instructions on how you can do this will come on at the end of Maria's presentation. But then, without further ado, Maria, welcome. The floor is yours.
Thank you very much. Okay, so let me share this one. Okay, so good afternoon, good morning, or good evening for, different people around the world, for being here, and for, I'm more than happy to share our work and our data with all of you. So my talk is Plasmodium liver stage silently making a bank, and I hope that I can explain this a little bit further. Okay, so I work with an organism that is Plasmodium that has a parasitic lifestyle. So basically, it lives to the cost of us, and not only us, but us and the mosquito. So it has two different hosts, the humans or mammalians or any kind of vertebrate, in fact, and then also the mosquitoes. And there's a very complex life cycle. So it's transmitted. So one of the hosts, the definitive host, where in fact the parasite has sexual reproduction, occurs in the mosquito. But then when we encounter these mosquitoes and they have parasites in their salivary glands, these mosquitoes transmit these parasites that obligatory must then go to the liver. And within the liver, as we already mentioned, the parasite traverses several cells, but within the final cell, the parasite is able to multiply into thousands of new parasites that then go out, and then these new parasites infect red blood cells. And they infect these red blood cells. Instead of having one single passage as it is in the liver, in the blood, basically these parasites infect red blood cells every 24, 48, or 72 hours, depending on the Plasmodium species. But they infect these red blood cells, they replicate in 20, 30 new parasites. These red blood cells burst, and then these new parasites will come and reinfect new red blood cells. So in fact, basically, malaria itself only occurs when this parasite is infecting these red blood cells and causes this massive number of parasites inside. Indeed, we know that very few parasites are deposited by the mosquito, but then when they reach the blood, they can be millions and millions of new parasites. The majority of the people are able to, be okay with it, but there is a proportion of people that, a huge inflammatory response takes over and then can be really lethal. we have done some work on these aspects, on the aspects of pathogenesis and aspects of that involve blood stage infection and involve malaria itself. But the vast majority of our group is really interested with one single question that is something that comes up with these numbers. Basically, we have a huge replication of the parasite occurring in one liver hepatocyte. So we know that the parasite, when really deposited by the mosquito in the skin, some parasites can never reach a blood vessel to reach the liver. So some of them get stuck in the skin and they can even start to replicate, but they give a kind of, maybe 10, 20 different parasites, each parasite. So probably there is not even enough to cause any kind of infection. We also know that, the parasites that come out of the liver when they infect red blood cells, I already mentioned to you, they can multiply in 20 or 30. But one parasite, when reached the hepatocyte and is able to invade properly this hepatocyte, this parasite gives rise to 10,000, 50,000 different parasites within one single cell. So really they all replicate within the same cytoplasm. There is never cellularization and division of the parasites until the last step, but one single parasite that we call sporozoite at this stage gives rise to thousands and thousands of infectious merozoites that then go to the blood and cause disease. The liver stage usually is called and is mentioned as to be the silent stage. And the reason why it's called silent stage is indeed because it's a stage that, the host never noticed that is infected. There are no symptoms associated with it. And basically malaria itself, the disease that Plasmodium causes, only appears when these parasites reach the blood stage and infect red blood cells. So we have been very much interested on this also because when you look in terms of evolution, we know that this is unique. When the Plasmodium species, there are many Plasmodium species that infect many different vertebrates, from birds to reptiles, and even one case of a malaria species that infect a fish. Basically, they have a pre-erythrocytic, so before going to the blood, they have this pre-erythrocytic stage, but only when there was the evolutionary jump of going to the mammalians, there was this liver replication and this huge replication. So this doesn't even happen when, for example, in birds, as here in chickens, where the parasite is deposited and infects some kind of, macrophage-like cells, but basically it replicates also probably into 50 different parasites. These thousands only happens within the liver. So what we have been questioning ourselves is really what you are observing here. What you see here is a liver section of a mouse that has been infected with Plasmodium berghei parasites, is a rodent species. And basically what you see is this liver section in a Z-stack, and you can see one parasite appearing here and one parasite appearing later on in this position here. And basically the parasite is able to do this. The hepatocyte needs to increase in volume 14 times and nothing seems to happen. So we are really very curious our question has been for the last 20 years, what hepatocytes really, and liver and hepatocytes, are so special. I should say that I have been at MIT in the laboratory of Sangeeta Bhatias in 2010, really trying to reproduce the conditions of a human liver or of a mouse liver in vitro, trying to have these parasites to replicate. But there is something special about an hepatocyte in a liver of a mammalian that is really something special that allows to reproduce in these thousands and thousands of parasites that doesn't occur in any other system. And in that respect, we have been approaching this question in many different aspects. Of course, we did sRNA screens, we did, microarrays a long time ago, we did lipidomics, we did proteomes. Many aspects are, and in every time that we have a different approach, we learn differently. The most recent thing that we have done is really we start to question if it was different for the parasite to end up in different types of hepatocytes. So it's well known for a long time that hepatocytes in these microlobules that the liver has formed, that they are very different if they are next to the portal artery or if they are more close to central vein. And we know that each of these hepatocytes is slightly different. And in fact, the laboratory of Shalala-Levitskovits at the Weizmann Institute in 2017 has published a beautiful paper that really have shown this zonation really by a single cell transcriptomics, and they have kind of a GPS from every single hepatocyte of each of these hepatocytes and really have different markers, so we know where it is. So when we saw this paper, basically I contacted Shalev and asked, can we really collaborate? We would love to really have this, your system, to know exactly where it is, and have two questions. One, is that, is it random, the hepatocyte that the parasite chooses to be in? And if it is, how is the outcome of that infection in one hepatocyte or the other? And basically, we have collaborated, and it took like 3-4 years to really produce the first spatial temporal single cell atlas of Plasmodium liver stage. And we say that is spatial and temporal because we did throughout the infection. And of course, it's spatial because, Shalev is this system that knows exactly which hepatocyte it is in. And basically, we answered the two questions that we have a long-term questions about it. Yeah, it seems that it's totally random where the parasite ends up. And we had this question because, as I mentioned to you, the parasite traverses several hepatocytes, to reach the final one. And so we were asking, is traverses because it's really looking for something specific? Doesn't seem to be. So really the parasite ends up in every types of hepatocytes. But later on, as the infection progresses, there is a much higher probability for the parasite to be successful and to really produce thousands and thousands of progeny if it's in a pericentral region, and there is a high probability of the infection to be aborted if it's in a periportal zonated area. We don't know exactly what is the reason for, and of course there is a lot of open questions that appears with this, and could be to the levels of oxygen and reactive oxygen species, could be millions of different hypotheses that we are testing now, but really gives us this feeling that yes, wherever the parasite ends up is not exactly the same. So the probability to really reach the next step or not is different. So also throughout the years, we have been interested on one particular organelle, we call it organelle, that basically is a membrane that surrounds the parasite. And basically what you are seeing in this image, this is in blue, is the nuclei of the whole cell of an hepatocyte. And basically this is the parasite replicating here, basically has been replicating now for, a few hours. So you already have a few, nuclei here. The parasite still only have one cytoplasm. But as you can see from the beginning, this parasite, when it starts to replicate, is surrounded by this membrane here, labeled in green. And this membrane is decorated by several proteins from the parasite and also from host origin. And we have been very much interested to understand why this parasitophorus vacuole is there important for whatever, and many hypotheses have been placed there. And throughout the past years, because we have started to really be very much and concentrated on this basically in 2014, 2015, and we have been producing several stories that shows different work. So yes, it is the parasite places a membrane here to block the autophagy machinery from the host. We also know that the parasite in this membrane interacts with all cell actin cytoskeleton, but there is a protein from the parasite that allows interaction but controls this. If it is not able to control it, basically the cytoskeleton, it really crashes the parasite. So we have been observing several aspects that have been already published. But one aspect that we have observed is that we have performed a screen for a lot of host molecules that are, in the venicities of the parasite. And basically, we realized that the parasites during the liver stages selectively divert distinct host cell compartments and becomes with a very proper endosomal signature that we are trying now to fully understand and see exactly the relevance of it. Okay. But the story that I want to tell you is more related to immunology. I should say that, you know, I'm not an immunologist, okay? I did a master's in immunology many years ago. I'm really very curious about these host-parasite interactions. You know, this is the aspect that really fascinates me and the people in my team. But, you know, we end up many times this obviously is a parasite, and we end up in immunology, and sometimes we need to learn with our colleagues here and friends to really answer the questions that we have. So within this question of why are livers and hepatocytes so special, there was always this kind of a dogma there that they must be really special, because not only as I mentioned, the hepatocyte needs to increase 14 times in volume to accumulate this usually replicating parasite inside, but also the majority of the times it was also, we don't see any kind of response around it. So as you can see here, and I've shown you several of these, you have this, you know, liver section here, the parasite is there, but besides having this huge parasite there, all the structure of the liver seems to be fully maintained. So in fact, the kind of word that we were using to you know, classify this stage, the silent stage, became not only that was no symptoms, but you know, the mosquito deposits so few parasites, only a few parasites arrive to the liver, that the parasite goes undetected. So there is a kind of a silent stage because there is nothing being detected there. But in fact, we know that this is not true, okay? Sometimes we see these things. They are rare, but in fact, they have been observed. Not that many people you know, have mentioned this, but in 91 was the first time that we could see a paper that shows a picture like this. Of course, it was not like this and colorful and beautiful, but shows the same type of thing. It was from the laboratory of Jerome Vandenberg at NYU that was doing a lot of microscopy, and has one picture of this. And basically, they didn't know exactly what to take off it, and you know, the dogma was so big that parasite will never be detected that they thought it could be an artifact because they were injecting a lot of parasites there, etc. But in fact, in 2005, the laboratory of Volker Osler started to quantify these, and they could reach they basically quantify these and reach the conclusion that could be at up to 10% of the infected hepatocytes would be surrounded by these infiltrates. Okay, we became interested on this for a complete different reason, because we were looking for host factors that were important to maintain the parasite. And in fact, we end up in one molecule that is called IMMOxygenase 1, and is an enzyme that degrades IM, okay, and deals with excess of IM. And basically, we realized that there was a kind of a lot of hepatocytes surrounding the infected hepatocyte that will produce express a lot of IMMOxygenase 1, and this seems to be blocking these infiltrates. And the reason why we know that is because we just tested and used a knockout mice. And of course, we were not expecting that it was infiltrates or anything. We just saw that the infected hepatocyte and surrounding hepatocytes have a high expression of this enzyme. And we used mice that were deficient in IMMOxygenase 1. And basically, we could see parasites. We could see pieces of parasites being destroyed by these huge infiltrates that look like almost like a kind of a granuloma. So then we thought, okay, is Plasmodium detected? Okay, we can see, of course, as mechanisms to block the type of detection, okay? So, but how is Plasmodium detected by the host? And so by then, there was not still single cell and all these type of things. We were still doing kind of a microarrays there. And there was already transcriptomics, and but we were at that time, we still did this work with microarrays, and we did, you know, the whole liver and infected and non-infected. And to be honest, when we had this, it was a postdoc in my lab, Peter Lille, that did this work, and he really wanted to do this experiment. And I was convinced that it was a completely stupid experiment, because the liver is kind of a sea, and the parasite is really something very small. It's like a needle in the haystack here, because, you know, even if we inject parasites that we take from salivary glands of the mosquitoes, we can inject maybe 100,000 sporozoites, these sporozoites, that is already a huge amount. But a mouse liver has 10 to the 9 hepatocytes and many more endothelial cells and Kupffer cells and everything. So I really thought, okay, we'll never see anything, you know. And I really thought if would have seen it, someone would have seen it before. But he insisted. And sometimes it's really good to have someone coming out of the field that insists to do an experiment that people in the field will think is so silly. And of course, nobody has done before, because people like me that were on parasitology, you know, although we think we are very, you know, non-dogmatic people, we already imagine, oh, this experiment will never work, we'll never do it. And he did it, and he could really found that there was a typical type 1 interferon response. So not only it was the only response that we could detect in the liver by that time, okay, in the microarrays, but when we used then, if not deficient mice deficient on type 1 interferon receptor, basically the response was all disappearing. And the interesting thing is that we could connect this to the infiltrates. Basically, we could show that in these mice, then we didn't have anymore this percentage of infected hepatocytes that had these infiltrates, okay? At that time, basically, we show in many different aspects, and I'm not going to this, but basically we show that somehow the hepatocyte, and this is not macrophage, this is not any other immune cell, is the hepatocyte where the response starts, and we have many experiments showing this, a lot of reviewers asking a lot of different experiments to show that this was the case. So somehow the responses seems to be detected in the infected hepatocyte. And as I told you, the parasite is surrounded by this membrane, this parasitophorus vacuole membrane, but somehow Plasmodium RNA reached the cytoplasm of hepatocytes is what we suspect. There is the involvement of MDA5, MAVs, and the transcription factors IRF3 and 7. But must be something else, because when we block IRF3 and 7, yes, we block the entire response. But if we block MDA5, we just block part of the response, okay? I should say that a colleague of mine, Stefan Kapp in Seattle, has shown that probably Plasmodium DNA also might be involved here. So must be, you know, different type of response at that time. For us, seems to be like Plasmodium RNA, and then the response is propagated. So even if you have very few infected hepatocytes, this response is propagated through the hepatocytes. And somehow then this call for kind of leukocytes, macrophages, neutrophils, etc., that comes to surround this infected hepatocyte. We don't know then, you know, we know that hemooxygenase 1 is involved then to avoid this thing. So, but we don't know exactly how these two things interact, but we know that parasite is detected. We finish with the dogma of, you know, more than 4 decades that indeed the parasite is detected, is recognized by innate immune response, and the response is mounted. Of course, the majority of the parasites still can make it, okay? And that is something interesting here. But this led us to the next question that we had. And this is, until now, what the field always will think is that, yes, the mosquito transmits, the liver just serves for the parasite to replicate, the parasite reaches the blood, and then when it is blood, it causes disease. The immune system recognizes there is a huge inflammatory response, and disease is caused by this huge replication of the parasite in the blood that, you know, leads to horrible inflammatory response. And so the majority of the people in the field working with mouse models, or even working with humans, they always associate disease with the blood stage, and blood stage being the only cause of disease. And so the models that they use is that they don't even use parasites coming out of mosquitoes that are very difficult to obtain and going through the liver, or etc. They have models that they use just for malaria models that they just do by blood transfusion. They transfer parasites from one mouse to the next through a blood transfusion, and this is it, is the models that we have ourselves. The stories that I told you that we have been working on blood stages, we use this type of models. But now we are just saying that, yes, prior to this, and in humans, this parasite is in the liver for at least 7 days, we know that the immune system has been recognizing. So we ask the question, does liver stage preconditions the host response to the next blood stage of infection? And so Angel Shora, a more senior researcher in my lab, basically decided to think, can we ask that? Of course, it's very difficult because if we start an infection with parasites coming out of mosquitoes, starting by blood transfusion, it's very difficult on the same day to have exactly the same number of parasites replicating in blood. All this is very difficult. So he came up with a model that basically, let me take here, but that basically he infects mice with parasites coming off the salivary glands of mosquitoes. He makes this infection, this liver stage, non-productive, basically by irradiating them or having parasites that are genetically deficient in a molecule that doesn't allow the parasite to come out, okay? And then on the same day, either on a group that has just blood stage or in a group that has the liver plus blood stage, he injects infected red blood cells. We always inject two different amounts of infected red blood cells because one is a little bit slower, the infection progression, the other one is faster, and we wanted to see what happened in both. And basically the result that we had, it was that the liver stage was really dictating severe malaria disease. So basically what you are seeing here is just survival here. And basically we have an infection in the blood just with blood stage, just by transfusing just infected red blood cells. And of course, if you transfuse a lot, 10 to 1 million infected blood cells, the mice really succumb much quickly than if you transfuse just 10,000 infected red blood cells. The parasitemias are accordingly. The parasitemia is the percentage of infected red blood cells in the blood. But interestingly, if we have now the model that we have previously infected with liver stage, yes, the ones that we call the liver stage, blood stage high, so liver stage and then high amount of blood stage, all the parasites still succumb. But interestingly and very surprisingly, basically if you have a liver stage but now blood stage low, all mice survived. And this is highly consistent, could not be explained by a difference in parasitemias. Of course, then parasitemias in these ones increase because basically they survive, but initially we could not see a difference on this. And this is not only for this model, that in this model, the mice succumb, and in malaria, people and mice succumb with different type of syndromes. And this is a neurological syndrome that we call cerebral malaria. But if we have an acute lung injury model or acute respiratory distress model, so the mice succumb due to respiratory distress, basically we have a very similar type of result. So then we ask exactly what was going on here. And I can tell you that I already showed you here the slide that is involved gamma delta T cells, but took us 3 years to reach gamma delta T cells. So Angel tried every kind of course, the first one was type 1 interferon because we thought it would be the one, is not. And then we try millions of different type of things. And then we end up that in fact our neighbors, Bruno Silva Santos is our neighbor here in the institute, a fantastic immunologist in gamma delta T cells, but took us 3 years to really knock in the door and test their mice. And indeed, you know, it really was, we were amazed, but we could not understand the result initially. So basically here, what you have here on this side is liver stage, blood stage low, that as I told you, you know, the vast majority of all the mice survive. And on here we have the other ones, the liver stage, blood stage high, that, you know, all mice or the vast majority of mice succumb to disease. But now when we do this, and this is when it happens in wild type mice, but now when we use gamma delta knockout mice, basically we have the ones that usually are protected, now they are not protected anymore, but the ones that really now, you know, they used to succumb to disease, now they are protected in the gamma delta knockout mice. And for me, it was really confusing for my brain because I thought, okay, how can in one case it protects and the other ones really, you know, is deleterious to the mice to not have these cells. And in fact, then talking to the team of Bruno Silva Santos, we realized that there are at least a dichotomy in the gamma delta T cells, there are many dichotomies, but one dichotomy that is either gamma delta T cells that produce interferon gamma or gamma delta T cells that produce IL-17. So we decided to test each of these, okay? And in fact, before we realized that when we really have these cells in the liver, the gamma delta T cells, we could realize that the ones that survive really produce very little number of cells that produce interferon gamma and a huge a number of cells or a high percentage of cells that produce IL-17, and opposite in the ones that succumb to disease. So we decided that maybe this is really the dichotomy that explains, these results. So in fact, we took advantage of some mice that have gamma delta T cells, but don't have the gamma delta T cells that produce interferon gamma. And when we have that, basically now, in the ones that are usually protected, they work as wild type mice, but in the ones that wild type mice are not protected anymore, now they are protected. So they work as gamma delta knockout mice. On the other hand, we don't have, and it's not possible to have, you are more immunologist than myself, but, we were never able to have mice that are deficient on IL-17 producing gamma delta T cells, but to use IL-17 deficient mice. And basically here now, the IL-17 knockout mice behave as gamma delta knockout mice in the ones that were protected, wild type was protected, but behaved basically as the wild type mice in the ones that were not protected. So it seems really this functional dichotomy of gamma delta T cell that really was explaining protection and succumbing to the disease, okay? And so basically what we have shown is that you have a liver stage that somehow really triggers a gamma delta T cell response, but then depending on high parasite mass or low parasite mass, basically you have more of one or other type. If you really you have triggered this gamma delta T cell response, but then very quickly have a high parasite mass, seems to really trigger these gamma delta interferon gamma producing cells. On the other hand, if you have a low parasite mass, you really have the gamma delta IL-17 producing cells. So two questions arise from here. Basically, how these protect, okay? And many questions arise from here, but the two that I'll focus. But can these really explain something that is happening in the field? High parasite mass versus low parasite mass, okay? And so basically by then we start to collaborate with Anne Tate, that is a professor at Vanderbilt University, and she's a mathematical modeler that likes to model interactions between cells, okay, cell-cell interactions, and she loves immune cell interactions. So we basically took our data and, can this explain anything of what is happening in the field? And basically what she comes up with, and she's not a malaria researcher at all, but she learned about it. And basically she thought that this could recapitulate what is being happening in the field after a few exposures. So what we know is that, in the field, in the malaria endemic areas, initially, when you are starting to be exposed to the malaria parasites, these children have a very high probability of developing severe disease. But if they don't die, basically with time, they never have sterile immunity, but they have a kind of partial immunity, and basically they develop mild and then, very mild infections throughout their lives. So the concept here, what she thought is that if you expose then, humans to these infection bites, and so the parasite is in the liver, triggers the gamma delta T cells that could somehow explain this. And by then, your the parasite mass in the blood basically decreases with time because you create a certain level of immunity. So initially you have a liver stage plus immediately a very high mass of parasites in the blood because you don't have this partial immunity, but with time, because you have this immunity, your parasite load is lower, so you would have like the liver stage, blood stage low as we have in mice. So having said this, the question that we were thinking, say, okay, but how this low parasite mass then is really protective? And we were very much interested in understanding this aspect. And again, First of all, we wanted to see if what Antate was telling us could be reproduced somehow in our rodent infections. And basically what we did, it was, instead of producing a liver infection with high numbers of parasites, we really had very few parasites taken from salivary glands of the mosquitoes, or even by mosquito bite, and produce, consequence like up to 4 times of these infections with low number of parasites, and then doing a proper infection. And as you could see, initially, if you just do one exposure to these parasites, yes, all mice succumb, but as you start to really expose them to these liver infections, basically the mice start to survive. And this could not be explained, the survival or not, by the difference in, huge difference in parasitemia by the time of death, but really could be saying by exposure to these was really creating a type of immune response. And then when you go to these to the proper infection, then you have a different type of outcome. You have a different outcome of infection. And the interesting thing that, again, a very long story, but taken very short, is that they seem to be dependent on the same mechanism as we were showing before. So it seems that indeed we would have a kind of an explanation of what is going on in the field. Although these are mice, obviously, and in a very controlled type of environment, obviously. So, but then we start thinking, so what is really the mechanism of protection? And so again, with the same system that we have a liver stage, and then 48 hours later, infect a low number of infected red blood cells or high numbers of infected red blood cells, and either using wild type or gamma delta knockout mice. And when you use, wild type, basically we start to see where were these gamma delta, cells, gamma delta T cells. And we look into many different organs, we look into the liver, they are very, very few. But where we saw that was a huge difference, and was a huge difference between the ones that are protected, these ones here, versus ones that were not protected, was a huge increase of gamma delta T cells in the spleen of the ones that are protected. So in fact, we could see that if we have different mice, wild type, gamma delta knockout mice, that basically develop that they are not protected, or IL-17 that they are protected with liver stage, blood stage low, we could see that the size of spleens were completely different. So we were really intrigued by this. So what we have done, it was just selectnomize these mice. And when we did this, when we have wild type mice that with this liver stage plus low parasite mass blood stage, that the majority of them are protected, if they don't have a spleen, they die more. So really, spleen seems to be critical for the protection, okay? So then we want to look exactly what is going on in the spleens of these protected mice in wild type, or mice that are not protected anymore, like either being the gamma delta knockout mice or the IL-17 knockout mice. And basically what we observe, and we obviously tested many different cell types, etc., but ones that really call our attention more was this huge erythropoiesis that was happening in wild type mice that were protected, and not in the mice that were not protected. So we thought, so is this the case? So what we have done was to understand the role of transferring either red blood cells, young red blood cells, or normocytes, normal red blood cells, into mice throughout infection in the condition of having liver stage plus low blood stage, okay? That usually they are protected, okay? And so basically what we have done, this is that in wild type they are protected, but if you use gamma delta knockout mice or IL-17 knockout mice, they are not protected anymore. And so we use, and this is one experiment with gamma delta, the experiment here with IL-17, and if we transfer normal sites, normal red blood cells, we give a transfusion with the same number of red blood cells, no infected red blood cells, but a transfusion of red blood cells, the mice still succumb as if we were not transferring anything. But if we transfer young red blood cells, reticulocytes, the majority of the parasites of the mice can survive. So really seems that basically what the mechanism that we are operating here is something that, you know, the spleen in the protective mechanism in gamma delta IL-17 producing cells is able to go through a huge reticulocytosis that is protecting these mice from dying. And of course, we have many players here, so this is really quite complex. We have a liver stage and a blood stage. The liver stage needs to be responsible for really the triggering of this gamma delta T cell. Then there is a dichotomy, depending if you have a low or high parasite mass, okay? And this is between the type of gamma delta T cells that you have and whatever they produce. And then if you really have, and dichotomy goes towards these gamma delta IL-17 producing cells, basically there is a huge reticulocytemia, and this seems to be producing. So again, before we have finished with this concept that the liver stage was not silent and was being detected by the immune system, but now we go further and show that in fact the outcome of infection, the outcome of a malaria as a disease does not depend on in the blood stage of infection, and depends really in a fine-tuning, in a kind of a balance between the liver stage of infection and the blood stages, okay? And of course, this really produces more questions. Any kind of work of scientific work produces more questions. And next questions that we are trying to work on is what triggers really the gamma delta T cell response. And this goes now to the beginning for work that we have done 20, 30 years ago, because we really think that, you know, many aspects that occur at very early stages of infection are really important for this triggering of gamma delta T cell response, and we are working on it. And of course, it really now leads also to other aspects of pathogenesis and work that in fact probably has nothing to do with the liver stage itself, because we know that even if we transfer reticulocytes in models that we have just blood stage going on, the reticulocytes are highly protective, and we are trying to understand how reticulocytes protect from severe malaria. And just to finish, to thank you all for listening to my talk, and also to thank all my team. I should say that this was made during the pandemic, but one day we had a retreat that could be only outside in 2021, and we were doing this retreat, and many of these people now already moved on, but they participate in this work. I think I mentioned all the people and the work that they have done, and our collaborators here, and of course our funders. Thank you very much.
Wow, that was amazing. Thank you so much, Maria. Your work is truly creative, and really the precision with which you've looked at this is amazing. And so that concludes our talk. But as I mentioned, there will not be a formal Q&A, but if you have questions, please feel free to reach out to Maria. The instructions on how you can do this via Twitter is there on this. So search for Global IMMUNOTALKS, and then find a tweet that says, ask questions for Dr. Maria Mota here, and then reply to that tweet with your question and mention global, #global immuno. So that concludes our session. I'd like to thank Maria very much for her wonderful presentation, and I wish you all well, and see you next week.
Thank you very much.
Bye.
Thank you. Bye. Thank you.